So I want to talk to you about some really almost embarrassingly elementary methods for looking at population genetic data, especially in humans, and some of the results that we've generated. But since this is an audience for physicists, I have tried to concentrate a little bit more on methods than the results, because I think my experience is that physicists like to get under the hood a little bit and see what's really going on. The thing I'm really trying to do in my academic life is try and understand human genetic history, and I'll make just a couple of comments on that. We got into this indirectly through medical genetics, and I'm not going to say any much about that route, though you can certainly talk to me afterwards about it. But our initial idea, which other people have had, is we would collect data from humans now alive. We would look at the structure, and we would back up from the current human structure and try and work out how it got that way. And we'd learn about genetic history. And to some extent, we still do that, of course. But our world kind of changed. And it changed for a technological reason, which is that in the last decade, it has become possible semi-routinely to extract DNA from ancient fossils. So increasingly now, we aren't looking at data of people alive. We're looking at people who are long dead and directly understanding the genetics of that structure back in time. And that is a technological advance that I had nothing to do with. It's wet lab technique. But it's really changed my world. And in the lab I work in with David Reich in Harvard, we currently have a database involving something like 7,000 ancient humans, and we are adding to that database at a rate of something like 2,000 to 3,000 fossils a year. Right? That keeps us pretty busy, as you might imagine. Anyway, I want to talk a little bit about some basic mathematical genetics and the natural model for phylogenetics, which is how populations and species separate, are tree-like models. But trees are not enough. Trees don't work at all well for modern human structure. I want to show you a formal test for whether trees work. I'm going to show you an application of that to the Neanderthal genome, which was a pretty remarkable piece of discovery at the time we did it. I want to talk a little bit about ancient DNA and some results on admixture. Admixture is when two distinct populations come together, mate, and produce an admixed progeny. And I want to give an example about the ancient history of Europe. And right at the end, I want to talk about methods for trying to date admixture, because we have tests we can very readily tell that a population is admixed, but it's not always so easy to tell when that happened. And let me say, I've been a noisy guy in these talks so far, and I would, if you dish it out, you got to take it. I'm very, very, I'm very happy if people interrupt and within reason throw fruit at me. All right. So I want to begin with some mathematical preliminaries, and in the spirit of the talk we just heard from Michael. I want to say that the entirety of this talk is based on the neutral model. I'm completely ignoring selection, and that's actually fairly reasonable in context here. The reason is that there's two reasons. First of all, I'm looking at modern human diversity, which from a genetics perspective is rather recent events. Let me just give you a little bit of background on that. Take two pieces of DNA which align, a tiny piece of my chromosome and a tiny piece of your chromosome that correspond to the same place in the genome. And those two pieces would have a common ancestor back in time. And we could ask ourselves what the distribution of that time back to the common ancestor is, and in particular what the mean is. So what's the mean distance back in time? And the answer is the mean time is something like 600,000 years, and 1,000,000 years back in time would not be remotely unusual. That's actually really remarkably old number. The modern human species is probably no more than 250,000 years old. So what that means is that all of you, are walking around with lots of pre-human mutations. You can tell your partner that. Right. And so the diversity of modern humans is actually quite recent on that kind of time. I mean, I would say a sort of natural time scale for human diversity in some sense is 1,000,000 years. But if we look at differences between human populations, they certainly diverge much more recently than that. So, and secondly, I'm always computing genome-wide statistical averages. So if there's stuff about selection, most of the time, with a little luck, it'll contribute with both signs as we sort of go across the genome, and it kind of washes out. Anyway, by and large, these models work extremely well on real data. So I'm not very worried about the neutrality assumption. OK, so let's do just a little bit of mathematics very quickly. So the first thing is I've got a population and descendant population y. That's what this arrow means. So there's a bunch of people back in time, x, right? They mate among themselves, and now we have a modern population y. And the allele, we'll look at some particular allele, and the frequency is a little x and a little y. So very important mathematical fact is that which was implicit in the Wright-Fisher model that Michael briefly described, is that the allele frequencies drift as a martingale. So in particular, the expected value of y conditioned on x is just x. Right. That rather elementary fact, it turns out to be extremely useful and important. And so let's slightly generalize this. We now have an ancestral population X, and it splits. And so there's two descendant populations now that aren't genetically interacting. The allele frequencies are what are X, Y, Z. And now, well, expected value of y conditioned on x is x. That's the same as before, and for z, the same. But also, the change in frequency between x and y and x and z are conditionally independent. They're conditioned on the frequency x. There's no dependency on what happens on y and z. And these drifts are uncorrelated and conditionally independent. I've made a lot of money on this slide, though. It seems it isn't very deep, but it is very important. Hello, sorry? Well, let's think about that. First of all, I mean, if you're looking at real, let's imagine we're looking at a real, we're looking at some ancient population. We're looking at, I don't know, British hunter-gatherers, right? Back in 6000 BC. And we're looking at modern English and modern Scots. Right? Then this model actually is completely false. But what can you worry about? You could worry about, well, have other people migrated into Britain? In which case they wouldn't be, the descendants wouldn't be there, right? Could there be mating between Y and Z? I mean, Scots and English sometimes don't like each other, but they do actually intermarry. You know, right, or there could be selection. You know, if there was genome-wide selection in England but not in Scotland, that would mess you up. And you know, like every other physical model, there's all sorts of assumptions implicit in here, and you have to think about that a little bit, and also check whether predictions of the model are matching at all well with the actual data. You know, that's the game. All right, so I'm going to tell you about a thing we call the F4 test. I'm going to tell you about a thing we call the F3 test. I'm going to tell you just very quickly about fitting a full phylogeny. There's a paper that I and my colleagues wrote, 2012, which if you care about this stuff, you can read. It's a methods paper. It lays this stuff out in some detail. And I think it does have some interesting examples, one or two of which I'm going to talk about here. And if you really want to get into the hardcore, there's a software package. It's on the David Reich's lab page, and you can download and run it. So, and plenty of data that's suitable for the package is available. So I'm also going to talk about dating, which is a different program and not in this package too. And the package has continued to grow. We keep on, this turns out to be a surprisingly rich idea, and we keep on thinking of new tricks. And then there's a new program we have to write. So, all right. So let's talk about testing, testing for trees. So what I'm interested in here is I have a whole bunch of populations, and what I want to know is there a phylogenetic tree that will explain the genetic structure that I see? So here's how this works. Let's just look at 4 populations. We may have a much larger set, and if we have a larger set, we'll apply this to every set of 4 populations in our set. And so you've got A, B, C, D, and the allele frequency is a little a, little b, little c, d, d. So I define an F4 statistic to be the expected value of A minus B times C minus D. And that's an F, so I said that's an F statistic, and A, B, and C and D needn't all be different. So this seems rather boring, but it turns out to be really very important, because the nature of what happens changes completely depending on how many of these things are the same. So if all 4 of these things are the same, that's called an F4. If 2 are repeated and the other 2 are different, that's an F3, and you probably can guess what an F2 is. All right. And there's only one unlabeled tree with 4 leaves. So that's an elementary piece of combinatorics, and I wrote down the tree. So if I am interested in labeled trees with 4 leaves, there's just 3 possibilities. And you can see that immediately. I can put A on the top left by symmetry. A has a friend, B, C, or D. There's 3 possible friends he can have. And after you've picked A's friend, the other 2 are fixed by symmetry. All right. So what I'm going to do for each of those 3 possibilities, I'm going to compute the appropriate F4 statistic by averaging over many, many places in the genome and looking at an empirical average of the population frequency. So I'm going to compute A minus B times C minus D at one place in the genome, and then another place. I'm going to add them all up. Now, let me just mention that I'm going perhaps a bit too fast here for some of this audience. What we're mostly doing here in genetics, and it's probably about 95% of genetics on actual mammalian data is we're looking at biallelic SNPs. So what we're doing is we're looking at places in the genome which are polymorphic in humans. They vary. And there's just two possibilities for what happens. That's the biallelic. Now, a remarkable fact, it goes because the human genome, as Michael talked about mathematical situations where you had a large number of positions in the genome and a low mutation rate, right? That's a very good model for human data. And if in the limit, no mutation will occur twice in a given position, and so you must have a biallelic SNP there, right? To get three possibilities, you have to have more than one mutation. Right? So, these SNPs, Michael even gave me the heterozygosity number, but 1 in 1,000. Human genomes, 3 billion bases. So they're going to be, between two of us, there'll be about 3 million polymorphisms. And overall, over the set of humans, there's something more than 10 million, we think, right? Maybe much more than that if you go down to places where the frequency of the mutation is vanishingly small. Right? I'm not really interested in places where SNP where there's one human being who has that allele, though there are no doubt plenty of those. I'd just like to put in a little pitch, which I think this is perhaps a good time. If you are a physicist or mathematician and you are thinking about moving into biology, genetics is an awfully good place to go. So this is like a sales pitch. And the reason is that the underlying models are extremely close to biological truth, right? They're not exact, and for most of the models I'm writing down, I can tell you the differences, but by and large, the differences don't actually matter much. They don't even matter very much to biologists, right? So there's some underlying truth there. And if you're doing some other things like systems biology, and you're talking about how genes interact and so on, that's extremely important biology, but it's actually quite hard to model mathematically. The mathematical models don't really correspond to the biology, or it's very difficult to make them correspond. And so sometimes you build mathematical machinery, and to me it's very unclear what the actual biological consequences are, because you know that the models are seriously off. And these models are off, but not by so much, I think. So anyway, that's my pitch, and that's one of the reasons I'm a geneticist. So anyway, I said we have three labeled trees, and we'll try them all. So if we take this first tree, A, B, C, D, then the mean of A minus B times C minus D should be 0. And similarly for the other two. And we carry this out slightly more precisely by computing what I call a D statistic. So here's how that works. I look at 4 alleles. I look at an allele in A, an allele in B, allele in C, allele in D. And I'm interested in situations where A and D match, B and C match, and A and B are different. Right. And I call that an Aber event. That's because you've got A, B, 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 A is the pattern. Right. And if you understand what an Aber event is, it should be within your mental powers to figure out what a Baber event is. Right. So the F4 statistic, in effect, says that the number of Baber events should be the same as the number of ABBA events up to statistical noise. And it's useful to put in this denominator. And it turns out that actually we're computing an F4 statistic by computing an empirical mean of the number of BABA minus the number of ABBA events. And so we can estimate D from sample allele frequencies. And one of the nice things about having a fairly big genome, which as a human I'm lucky enough to have, is that really the standard error of these numbers turns out to be quite small. So this test has actually got real power. So here's some sample results. So a big project initiated by Luca Cavalli-Sforza, who was at Stanford at the time in the 90s, was to collect samples from a multiple set of diverse human populations. And these samples are immortalized because we have cell cultures currently held in Paris. This is called the Human Genome Diversity Project. And here are some results from that. So Yoruba are a group in Nigeria. San are the South African hunter-gatherers, Han Chinese, and a North European population. And so we compute our D statistic, and the Z-score is minus 0.56. Totally boring. And you try the other two scores, and you get 78 and 80 sigma. Those Z-scores sort of tell you just how powerful this test is for figuring out that really Yoruba and Han are not a clade with these other populations. So this is okay. It's telling you that two actually highly diverse African populations and two diverse Eurasian populations fit this tree model. Okay, that's right. So now let's replace our Bushman population with individuals from the highlands of Papua New Guinea. And you run the same stuff, and all three trees fail miserably. Right. And this happens actually more common than not. And the historical reason is, of course, very clear, which is that over and over again, human populations don't just split and not mate. The splits and mergers are very complicated. Populations split off, and then some subpopulation migrates, and so on. And these are very interesting historical events to understand, but they show that the tree model, when that happens, the tree model is just seriously wrong, and these statistics will tell you that. So in general, the history of Eurasia, and in fact increasingly we're learning the history of Africa, are very complicated. And my job is to really try and understand that complexity as far as I can. Try and unravel what actually is the sort of simplest story that the data doesn't falsify. Yes, these statistics you take, actually, well, being very precise, we do this on the order zones. You've got 20, as you are humans, you each have 22 order zones, one X chromosome and another X chromosome if you're female and Y and the mitochondria, but we mostly do these statistics on order zones because the X and the clonal, the X and the mitochondria and the Y chromosome have extra issues. X is actually extremely important for understanding a lot of this stuff, but it's a little more complicated to analyze because you really have to deal with the male and female samples differently. Okay, so that depends on your purposes, and I think I didn't really tell you, I think I may have a slide somewhere about how the z-statistic is computed. It would be hopelessly naive to simply do these counts and assume that they're all independent, right, snip by snip. And Michael mentioned in passing this phenomenon called LD, which really is that two places in the genome which are very close together don't operate independently. And the reason is very simple, which is they share a genetic history, and unless you have a recombination event that separates the chromosome between those two alleles, they're going to share the same genetic history, and that introduces statistical dependence. And we see a great deal of that in humans. It depends on the population, what the width scale is. And also it depends, there's a statistical roll-off, so there's not exactly, you can't just cut it cleanly, but tens of thousands of bases is the right scale for Eurasians. For some populations which are deeply drifted or bottlenecked, it's much longer. And for, say, West Africans, which have had a larger population size for longer, it's a little less. So these statistics are computed with I'm quite good at stealing other people's ideas from other fields and claiming full credit. And a technique that I introduced in genetics is the block jackknife, where you divide the genome up into large enough blocks that the only real errors is dependencies near the boundaries. The default we use is approximately 5 million bases. And there's a known statistical technique for computing standard errors using data that you divided up into blocks and applying the jackknife. And that works extremely well. That's become a fairly pervasive technique in the field, in fact. Here's a few other examples. So Yoruba I talked about before. Adyghe is a modern population in the Caucasus. Dai are in South China. And the Ongi are a fascinating population, which I'm extremely lucky to have a genetic availability. That's a population from the Andaman Islands in the Indian Ocean. The Ongi live on Little Andaman. They're rather tragically a very small population size now, probably only about 100 adults. But why they're important to me, apart from their own intrinsic genetics, which are interesting in various ways, is we believe they've been genetically isolated for at least 10,000 and probably 20,000 years. So there have been humans on the Andamans for all that time with very, very little contact with the Eurasian mainland. And that gives us a genetic window back in time, really quite a long way. So anyway, you see from that first data, for these four populations, you can't get a good, something's going on. And I think what's happening is that after Ongi are an Asian population, but there has been some gene flow between the Far East and the Caucasus over the last 20,000 years, and that's been enough to mess up the simple F statistic. And here's another example where there's another population I mentioned later. Caritiana are an indigenous population from the Amazon rainforest, and we're lucky to have those two too. And that's technically they're very helpful to us Most populations in the Americas have admixture from colonial times. For example, there aren't any populations in there aren't any indigenous populations in North America without European admixture. And that causes some technical complexities to people like me. But the Caritiana are indigenous in the rainforest, and as far as I can tell, have absolutely no admixture. And so they're a kind of population that I can use as an example of indigenous American populations before Columbus. And I'll just make a comment on the last line. We have Chukchi, who are a population in the peninsula, the Russian peninsula, right by the Bering Sea. It's the northeastern point of Eurasia. Han Chippewas are an indigenous population from Canada, and Zapotec are from Mexico. And what this is reflecting, we believe, is back migration from the Americas into the European mainland. We believe that the Chukchi have picked up genetics from the Americas probably in the last 1,000 years. Human genetics is complicated. You get gene flow in all kinds of directions. Yeah. Oh yeah, I should say, yeah, sorry, this is so familiar to me. Thank you very much. I appreciate that. So, okay, let's take, let's see if we take any one of these examples. Let's take the Yoruba Adage Ongi dye. Okay. So that's 6 sigma, doesn't fit a tree model. So there's got to be gene flow between some of these populations. Plus means that there's gene flow between either the first and third or the second and fourth population, right? And minus means gene flow, you can guess, between the first and fourth or the second and third population, right? Now in this case, let's just do some common sense. Yoruba is from West Africa. We believe there's been very little gene flow between Africa and most of Eurasia in recent times. So gene flow between the Yoruba and the Andamans is not a good guess, right? Huh? So that means there's almost certainly the answer is there's gene flow between Adage and Dai, right? But what you can't tell with this statistic is which direction that flow went, or whether it's in both directionals. There's just something is making Adage and Dai closer together than they would be under a simple tree model. That's what you learn. And so the basic game I've been playing, or one of the games I play over the last 15 years, is to take this kind of statistic and see what we can actually learn about history. Right. So here's perhaps the most famous example. We look at some archaics. And the two archaics I'm talking about here are, I call Altai, which was a Neanderthal fossil found in a Siberian cave, and with extremely good genetics. We have so-called 50x coverage from Altai, which means that you have 50 independent reads on average overlapping each place in the genome, which gives you extreme. The quality of these two archaic samples, each more than 50,000 years old, is not significantly worse than if I took a blood sample from you and ran out the DNA in a good modern lab. It's remarkable. The state of preservation, quite remarkable. Denisovan is found actually confusingly in the same cave at a different layer. Um, Probably a 10-year-old or so girl, quite remarkable. All we have is a single joint of a little finger. This little joint here, right? From that joint, my colleagues in Leipzig in Germany, recovered more DNA than for all the Neanderthals that have ever been recovered. That finger bone is just packed with DNA. We don't understand why that happened, but we're lucky. Maybe I'll just tell you a little story about a colleague of mine. It just shows I could not possibly do this, but a very brilliant colleague and friend of mine, now running a major lab in Jena, Johannes Krause, was working on this finger bone, and all he had was he had a box of ancient bones, otherwise completely undistinguished, and he's systematically working through the box. And there was nothing even to indicate this was an archaic human. I mean, he thought probably he was looking at some modern human finger bone. And he gives a lab meeting talk, and it's full of frankly boring technical details. He talks for 40 minutes. These are the reagents I use. This is the temperature I ran the reactions at, blah, 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 blah. He's got 5 minutes left in his talk. And the average age of human mitochondria, in fact, the maximum age of human mitochondria is something like 150,000 years, right? And he says, and he finally says, deadpan, and the divergence of the mitochondria of this human I've just discovered from modern humans is more than 1 million years. So, that was just, that's totally mind-blowing, right? You are not looking at a modern human, but that's what's happening. And indeed, it turns out that Denisova is a sister group to Neanderthals, but moderately diverged from them. All right, anyway, so if you didn't know anything, you know that the Altai and Denisovans are archaics, not modern humans. Mabuti, modern humans from Central Africa, French. So you'd think that this is the tree. You've got chimpanzee, humans diverge, Mabuti and French are modern humans, Denisovan and Altai, that would have to be it. You run out, particularly the Altai, you look at chimp Altai, Mabuti French, the D statistic should say 0, it's 6 sigma. And that told us that there had to be gene flow between Altai-type Neanderthals and Eurasians that's different from gene flow in Africa. And further analysis showed that the gene flows in the direction of Neanderthals into Eurasians. I'm not going to go into that. I'll just make a comment on how we try really hard to be careful here. The standard paradigm, which you've heard from, I think, David Ping's talk yesterday, is you take short reads of DNA, Illumina reads, and align to the human reference sequence. Right? So we did that. But the human reference sequence, suddenly each local area of the human reference sequence is some human, right? So there's an intrinsic bias there, which is that the human had to come from some population. I mean, humans aren't all the same. So we were, this result was sufficiently important and sufficiently surprising that I was scared that some alignment error or systematics could be causing this, right? Maybe for some reason the Altai the French is closer to human reference than Mabuti Pygmy, which it is, and were aligning to the human reference, and that's causing some systematics. And we couldn't see how that could be working, but we were scared. So I actually made Svante Pääbo, the PI of the whole project, spend some significant money, as it was at the time, and we realigned our entire data set to chimpanzee instead of humans. And chimpanzee is, of course, moderately diverged, and you wind up with worse alignment. But the point is that all humans are equally diverged from chimpanzee. So there's no real way that bias to the chimpanzee alignment can be introducing population-dependent nasties, right? So we did all that, and the statistics didn't change. And I still think Svante may think I wasted his money, but I didn't. Yeah. Huh? Yeah. Huh? All right. Okay. So now I'm going to go on to the, well, you did the F4 test, so now I'm going to do the F3 test. And so here's a little tree. And we're now going to talk about F3 test, which is C minus A times C minus B. So C is coming in twice. Well, you think about this, and you see that with a simple tree, these F3 tests, these F3 statistics, all have to be positive. And actually, I did discover this thing, and if I was very, very smart, I would have worked out that you could get negative values for admixed populations. But it didn't work that way at all. I had a program that computed F4 statistics, and I thought one morning, I wonder what will happen if I compute F3s instead of F4s. And there's some minor problems. You have to think through, which I did, and because in effect, when you're computing this thing, you need to estimate C squared, and the sample frequency squared is not a good estimator of the population frequency squared because of sampling error. But you can figure out the statistics and work out an unbiased estimate for the actual population frequency. So I did that, and I ran out on a whole bunch of human populations, which I'm going to show you soon, and there were some massively negative numbers, which I was quite surprised at. And then I sat down and did some math and figured out what had to be happening. If I was Albert Einstein, who was actually the first member of IAS, an institute joke, which Arnie didn't tell, I was surprised, which I like, which I heard from an institute director, is that, yeah, Einstein was our first hire, and since then the quality's gone down. So, all right, anyway, so here's a different model, not a tree model, in which you have a population C prime, which is an admixture with mixing weights alpha and beta, and then there's drift from C prime to C. And when you think about this, it turns out that the expected F3 statistic, it has a term C minus C prime squared, which you kind of expect because that's the drift from the admixture event. And then there's the interesting term, which is alpha beta, the product of the beta is 1 minus alpha, of course. It's alpha beta times an F2 statistic between A prime and B prime. I'm not going to prove that to you, but if you're actually interested in this stuff, I recommend that as an exercise. Decent, it's not hard, and if you have any mathematical power, you'll prove this very quickly. Or if you really care, you can look up my 2012 paper and cheat. Right. To get a negative number here, this drift C minus C prime better be small, and you need alpha and beta can't be too close to 0 or 1, and finally the admixing populations have to be fairly divergent. All of that makes perfect sense. I mean, if alpha is very, very near 0, you've hardly got real admixture. If A prime and B prime aren't diverged, well, of course you're not going to see anything. Here's what you get again from the Human Genome Diversity Project. So I think I remember I ran this program out, and I sorted on the F3 statistics. This is the first time anyone ever done this, and I saw that, and I saw my most negative score was minus 73 sigma, and my immediate reaction is I must have screwed up the statistics, right? I mean, nothing's 73 sigma, but I hadn't. It really is, and this is the Uyghur population, which is a genetic mix of an East Asian and West Eurasian population. You can't tell. All you can tell from this is that the admixing populations should be genetically somewhat similar to these populations, right? Obviously, it's highly implausible that the French in Paris have admixed into Uyghurs in a massive way, but right, that's not what we're claiming. But it is true that a West Eurasian population has definitely done so, and this was known. The Hazara also came out. That's actually an interesting tale, which the Hazara are thought to be come from Genghis Khan's conquests. They're a population, they're a tribal group in Afghanistan, and what seems to have happened is Genghis Khan and his armies marched through Afghanistan, and he left behind troops as a garrison. And they then admixed with the local population, who are West Eurasians. And so again, you wind up with an East Asian population and West Eurasian. And for some reason, I get these weird Druze from the and Japanese. But you should again think West Eurasian, East Asian. And that's known. Xhosa, a large population, tribal population in South Africa, who have substantial admixture from San Bushman, which is also known. And in fact, there's linguistically, the San have a remarkable language with a number of, something like 20 click phonemes, these click languages. And Xhosa absorbed 3 of the clicks into their language. So both linguistically and genetically, there's known admixture. This is interesting. The two are an ethnic population in West China, and I looked them up on the Wikipedia, and their standard name from the surrounding people are white Mongols, and they're admixed, which I very clearly. And I want to talk about the last example, which really actually surprised me. This is Sardinia and Caritiana, who are this population from Brazil, appear to have mixed into modern French. And that was not so clear what that could mean, but I'm going to tell you. Okay. So first of all, I do not think that the modern French are a product of back migration from South America. If I did think that, you should probably call an ambulance, because. But here's what is happening, and which we figured out in the 2012 paper. We figured this out. So there's a population which we call a ghost population, because we're confident it existed, but it doesn't exist now, right? And it's simply been inferred from the modern data. And we call these ancient North Eurasians. And the two things happen. The ancient North Eurasians, or some of them, cross the Bering Straits and go into the Americas. Right? Eventually, with the Caritiana as a descendant population. Um, uh, I should say, earlier on, backing up, there was an out of Africa event where where the ancestors of all modern Eurasian populations leave Africa. Some of those people go into ancient Europe. Some of them become the ancient North Eurasians. And then we get an admixture event where some of these admixed populations form a population in Northern Europe, and also we think this long diagonal line here is actually quite a weak event, but there was some evidence in the data. But also ancient Europe can form into Sardinia. But this is a small event, and this is the large one. So fundamentally, we're seeing Northern Europeans are a mix of ancient Europe and ancient North Eurasians. And you can see then that this will give you these sort of correspond to my population. This is population C. This is population B. And these divergent events are invisible in the F3 statistic. That's one of the things that's remarkable about these stats, is that very often you've sort of simplified the phylogeny, and important events become invisible, which is in many respects good, because you can form statistics that are in some sense revealing a simpler truth anciently, right? So this diagram appeared in my 2012 paper. Yeah.
You found this based on an F statistic with respect to the Sardinian and one particular American population.
Yes. Now the other populations all work, but but if you put in a population that's heavily European, a modern population with heavy post-colonial admixture, that dilutes the statistic, right? Just as you would expect, right? I mean, it's true. No, the Caridiana, the other populations for which this also works, and in fact, even if you replace the Caridiana with Han Chinese, you get a statistically significant statistic, and we now think the reason for this is there's been gene flow between the ANE and the Han, right? But the Han are a whole another subject with their own unique and complex history, much of which has nothing to do with this event, right? Just wait a little, right? Yeah. Yeah.
When you have someone from an unusual population, the sampling usually is in an individual or two individuals.
It's as many as in the Caridiana it was like 10. And actually that brings me on to a point I wanted to make. No, well, yeah, no, yes, that's what I'm going to say. So our work has nice tune. I think it's genuinely true that this ancient DNA work is transforming the archaeology. We have a whole new route for looking at things, and I have many wonderful archaeological colleagues who are really happy to be contributing to this. There are other archaeologists who are not so happy. And the reason is that we are refuting long-held theories. And one of the criticisms we've been made is that we're looking not so much at the modern DNA, there are issues there, but we're looking at a handful of ancient fossils and making broad conclusions. It's worth making, so what to say about that? Well, first of all, it actually makes sense, in a real sense, to be doing serious statistics with standard errors with a sample size of 1. And the reason is that if you have a single individual, as you run across the genome, you are in fact sampling you're sampling thousands of their ancestors, right? Now, you obviously have to worry that your sample is not representative of the true population. That's a worry. But if you have as few as half a dozen samples, right, all of them show the same features, right, and you're all looking at thousands of ancestors from each of those, the worries become pretty thin on the ground. And an example of, specific example, which many people here will know, about a dozen years ago, a remarkable find was the so-called Iceman, who was exposed on a glacier in the Alps on the border of modern Italy and Austria. And the Iceman genetics, which were quite easy, it was amazing preservation, easy to get DNA. The Iceman resembled modern Sardinians. And it was hard to know how to interpret that at the time. One worry I had was that, this is a guy whose parents maybe migrated from Sardinia, right? And that was why he looked like a Sardinian. But it's turned out since we know much, much more about the genetic history of ancient Europe. His genetics are absolutely typical of the people living in his time and place. Yeah.
Example of what I think is in the literature. You'll know this much better than I.
Yeah.
There were mating between Neanderthals and other quasi-humans, humanoids, in this 800,000-year period that all these organisms, the old ones, have lived. There was a report, I think. I actually just read about the report. Unfortunately, I didn't read the paper. That the mother was the same in 2 examples of these matings, and that in other words, the question of viable breeding between some of these older examples.
Yeah, there's some. I mean, you have to. But of course, the inbreeding. If you have a sample, inbreeding is trivial to detect, and we worry about that. But in general, I cannot think of a single example in my ancient DNA world where we've had 1 or 2 samples to begin with, we've reached wrong conclusions, but which became clearly wrong when we got more samples. I'm actually that no doubt will happen. Sooner or later, we're going to get some ancient sample that was very atypical of his population group. But yeah, and for the Cavadiana, we have 10 samples and they're homogeneous and everything's fine. Oh, we can now. We can now. We have. No. But these aren't random Sardinians. They're a population. Luca, actually, I mean, the 23 Sardinians we're using, and actually there's a variety in Sardinia. The statistics. Well, you would get a weaker statistic because you can tell. It turns out actually there's a slight digression. Actually, every population geneticist finds this the hard way. They don't really believe if you're a physicist, you think that you have standard errors with a sample of 10, right? I'll go to a sample of 40, and my standard errors will halve, right? And if I go to and keep on doing that, everything will be great. And everybody thinks that, even though they've been told it's wrong, and they find out it's wrong. And the reason is that the limitations we have are that the genome is not infinitely big. You can only sample so many regions of the genome right? And after a while, increasing the number of samples of human samples doesn't help. There's intrinsic variation in what happens as you run across the genome, and you could take 1 million Sardinians, it wouldn't actually drive down your standard errors very much. That's just the way life is. God should have made a larger genome, but he didn't. All right. So here's what actually happened. I gave you the F statistic, and here's what we now understand, which I forgot my punchline. I got to get my punchline. So we published this paper, and the year after we published this paper, another group find a 24,000-year-old fossil in Siberia. He was a 7-year-old boy, and he exactly matches the genetics we want, right? So in other words, this ghost population stopped being a ghost. The ghost showed up, right? So, all right. So here's what happened in the history of Europe. So the ANE are living in Siberia very anciently, and they contribute to later Siberian genetics. And then the Siberians' genetics moves on to the Russian steppe. We're still sort of arguing a little bit, but in this sort of time frame of like 6,000 BC, we get Siberian genetics on the Russian steppe. And around 3000 BC, time of the Iceman actually, Europe has no Siberian-related gene flow. But the steppe but there are pastoralists on the steppe. It's a culture very well known to archaeologists called the Yamnaya. And the Yamnaya pastoralists in 3000 BC have tons of this Siberian-related ancestry. And then there's massive immigration from the steppe into Europe. Happens between 2500 and 3000 BC. There's a culture all over Central and Eastern Europe called the corded ware, which is something like 70% of the ancestry is steppe. And this steppe ancestry then moves further on into northwestern Europe, into Britain, and a little later into Iberia. And is in fact the most recent major event that leads to modern European genetics. So all Europeans today have some steppe ancestry, with more in northern Europe than in southern Europe, and that's driven by this Yamna migration event. It's off topic here, but I'm convinced that the Yamnaya spoke an Indo-European language and have contributed genetics to every modern Indo-European population, both including modern India. There's a paper that has now been accepted in science that's a monster paper on the history of Central Asia and South Asia, and if you look out for that, if you are interested in this stuff, there'll be some news publicity about that, I think. I had a question from Michael, I think, about trying to look a little larger at phylogeny, and I don't want to go into too much time, but you can build a model like this. This is, we call this an admixture graph, with, in principle, an arbitrary number of these admixture events, and the solid edges here are drift events, with the numbers indicating how much genetic drift you have going down the edges. Right. So you wind up with a parametric model from this, and given such a model, we can calculate theoretically the expected value of any F statistic. And we also can compute an error covariance. So that makes it possible to compute an approximate likelihood. You take your data, you take this model, and you compute the joint distribution, empirical you compute the empirical distribution of all your F statistics, and you have a model because you have the covariance of what that joint distribution ought to look like, and you get a test. The details, this has proved quite effective. I mean, we have a program that does this. It's proved pretty powerful, but there's lots of problems. I'm quite dissatisfied. I don't really know what the score I should be using for the fit is. I don't really know how to fit the graph. I have a method. It's a little clunky. I badly need a goodness of fit test. You know, you have this score, you fit a whole bunch of parameters, like it's quite a complicated parametric model. There's questions, how many parameters have I got? Well, you might think that's a trivial matter. I mean, you just count how many, numbers you had to fit. But in fact, I mean, you get things like you have an admixture event where it's detectably non-zero. You're sure of that. But the admixture is small. It's like 2%. The standard errors are significantly, the standard errors matter. And is that 2% a real parameter? I mean, it isn't affecting most of the F statistics. So I don't have a good fitness of I don't have a goodness of fit test, though I have some ideas. I would really like a better one. And as an important practical matter, we really ought to need a better way of building large graphs, right? If you have we're building a mixture graph sometimes with 15 or 20 populations. There's a lot of potential graphs out there. I can't try them all. And I need much, much better methods for building these graphs automatically. I have two colleagues who are working on this, and they've made progress, but they sure haven't solved the problem. So if you're interested in this kind of thing, come and talk to me more, and I will be happy to try and give you some examples. So I'm gonna start winding up, though I'm still got a little time. I want to tell you about one more problem in this general area, which is dating admixture. I mean, we have an admixture event. Maybe you have an F3 statistic that's way negative. We're quite sure this population is admixed. We'd like to know what the source populations were. That's for sure. But we'd also like to know the date. I mean, if you tell a historian, well, I'm sure this population is admixed, but the admixture is between 1,000 and 4,000 BC, he isn't actually going to want to hear you much more, because a lot happens in those 3,000 years, and that's not useful information for him. So we need to date. Well, I'm not going to go into the here is we really actually need help on this, and I thought I'd show you at least an example of where it's working well. So I already mentioned the Yamna. We have more than 30 samples from a Yamna settlement in central Russia called Samara, and I built sources from the ancient steppe in Russia, like 5,000 BC, much of which is in fact before farming or pastoralism, and also from Iran and ancient Iran and the Caucasus. It's pretty clear to me that whatever's going on, the Yamna form is an admixture from some populations that are coming up through the Caucasus that are genetically Iranian, and mixing with people who are already there. So you wind up with this I'm going to tell you in the next slide exactly what we're looking here. But you wind up with this curve that's an exponential curve, and the decay rate of the curve allows you to read off the admixture date. And the admixture date is, of course, relative to the samples that you're looking at here. So the Yamna samples date from about 2900 BC, right? You look at this decay curve, it's about 50 generations. Rule of thumb is like 29 or 30 years per generation. So we're talking roughly 1500 years. So this is 2900 BC plus 1500, that's about 4400 BC. And you can see that the curve is obviously well determined from the data, so the errors on that aren't going to be very large. So how do we do that? Well, the basic idea is clear and probably a good one. The details I'm still struggling with. The central idea is to actually exploit LD. So I look at two places in the genome and I look at a chromosome from ancient Yamna, right? And so I got a left allele and I got a right allele on that chromosome. Well, there's two possibilities. They can have come from the same admixing source or different. And you can tell that if a recombination event has occurred in between the admixture event and the chromosome you're looking at, it's independent. The left and right sources are now chosen independently. But if if no recombination event has happened, then the left and right have to have been taken from the same source. So what I do is I model the genotype across the genome as a mix of my two sources, and I compute the mixing proportions. So I just do this by stupid least squares, easy peasy, and I can estimate the mixing proportions, which I do, and I can compute a residual. And we also, on this SNP, can get a weight, which is the difference of the allele frequencies of the two sources. And now it turns out that you just form the product. You take this weight times the residual, and if it's positive, it's more likely that that your genotype came from the A population, if it's negative, from the B. So the correlation function of X should be exactly telling you how quickly you decay off into independence. Right? So this is, physicists tend to call this sort of stuff a two-point method, and that's exactly what I'm doing. I'm doing this for every pair of SNPs throughout the genome and averaging the statistics by genetic distance. It's not at all clear this is the best answer. I mean, there's the idea here, there's got the exponential breakdown, but exactly what the stats should be, I still don't know. I'm still struggling with it. We have a set of quite powerful techniques here. It's uncovering a huge amount of history. It's repeatedly showing some huge surprises. I'll just mention one, which is, I think, quite remarkable. I mean, I like to say that I study the genetics of ancient Britain as well as the stuff, and I like to remark I'm an ancient Briton studying ancient Britain. So here's a huge surprise to British geneticists. They knew farming starts in Britain like 3800 BC, and get Stonehenge, the famous site, built about 2800 BC, and a few hundred years later, you see a culture which was well known on the mainland at that time called the Bell Beakers. So Bell Beakers show up in Britain like 2300 BC or something like this, and then later on it gets to the Bronze Age. And the archaeologists thought that the Bell Beakers was just a cultural importation from the mainland. that there's trading going on between Britain and mainland Europe, and they see bell beaker culture and they learn how to do bell beaker stuff in Britain. Okay, we look at the genetics. Between 2800 and 2300 BC, there is a 90% population replacement. The bell beakers have almost nothing to do genetically with the people who were in Britain just a few hundred years before. And there's no doubt about that, and it was, came as a complete shock to British archaeologists, many of whom still don't believe me. This is a huge effort. David Reich, my colleague, contributed massively. I just mentioned a few. Nadine Rowland is a amazing lady who set up a fantastic wet lab. These are Sharp and Joseph, are two of my closest colleagues. Priya Mujani works with me on dating. We're still struggling and trying to improve what we're doing. All the Neanderthal work was driven by Svante Pääbo. We of course rely on the archaeologists and the individuals, ancient and modern, who provided the data. But before I pack up, I wanted to, I have no actual, this is totally unconnected with my talk, but I like to do it anyway. Here is a fantastic picture, which I like to show whenever I can. This is as you can see, this is a horse. It's carved from mammoth ivory. It's 30,000 years old. And for me, this is not science, but this says these ancient peoples were not primitive in any real sense, right? Whoever did this is a real artist, right? Somehow he is my brother or sister. I really feel that very deeply. I've seen this fossil. It's very small. You can hold it in the, it's in a museum in Tübingen in Germany. They wouldn't let me do it, but you could easily hold it in the palm of your hand. But isn't it beautiful? All right, I thank you very much. Oh, good. Yeah. Sure. Yes, of course. Of course, they have pot. That's what they got.
Do you have a sense from the genetics which artifacts are good to date based on, and which artifact histories correlate well with genetic histories?
I don't. I think funny question. I don't think we have. I think actually you don't have much choice. I mean, ceramics are much the most common thing used because they're well preserved, right? Some of them are valuable, but some are not. I mean, you know yourself. You have a coffee cup. It lasts a few years. You drop it, it goes in the trash, right? The ancient people did the same. Yeah, yeah. And I do think that we're seeing you see genetic continuity, and you also see genetic breaks, and it's very hard to guess. I mean, as an example, we think farming came into Europe quite clearly with the movement of people. Farming in Anatolia seems to have been derived from the Fertile Crescent with no genetic movement. Pastoralism arises in Iran. Again, it seems to not have involved genetic movement. So sometimes yes, sometimes no. And I don't think it's easy to tell from the archaeological objects which of those cases you're in. Yes, I'm interested in that, and to the extent that's uncontroversial. But here's a very important example. There's a Indo-European group called the Tocharians. Tocharian was spoken in Western China in the first millennium BC. And we know that because they were literate. They have an Indo-European language written phonetically in Chinese characters, and we can read it. We think that they are descendants of a population called the Afanasyevo, which moved to Siberia about 3000 BC from the Yamna. And again, you have Indo-European language, and we know that some of these people in Western China in first centuries BC are in fact genetically West Eurasian or have a major West Eurasian component, but sometimes not. And the linguists are still arguing about that. I'm not a linguistic specialist, and I think linguistics can be a clue about the genetics, but you can't expect a perfect match. All right, I don't want to delay lunch anymore. I'm sure you're hungry.
One more question, and then I get the impression that the ancient DNA that we identify as Neanderthal is largely from sites in Siberia.
There's a magic cave. Yes, there's a magic. Yes, there's a second magic cave in Croatia. We have two super high quality Neanderthal genomes now. One, the Altaj I mentioned, the other from Vindia Cave in Croatia. And we have a number of other Neanderthal fossils where we have useful DNA, but not of the same quality. I think you just need a lot of luck. The ideal conditions are a cold, dry cave, and it needs to be cold and dry for tens of thousands of years. And that's unusual, but not impossible. Right. Yes?
So I don't study ancient DNA or evolutionary history at all, so I'm very new to this. It's very interesting talk. But I skimmed recently on Nature that there was recent improvements in looking at protein, ancient protein.
Yeah.
And I thought that was really interesting. I was wondering if there's been a lot of new analysis showing that, or somehow related to these populations and ancient populations.
No, that's very, the ancient proteomics is really cutting edge. Our lab isn't doing that. The story there is that protein is preserved, DNA is preserved for hundreds of thousands of years. Proteins can be preserved for millions of years, but only a few proteins. Collagen in bone and so on, that's good, right? But you're not going to get blood, no luck. The problem from my point of view is also that protein evolves very, very slowly. So I think they found a jawbone in Tibet, which is like 200,000 years old. And if I remember, they found one mutation in the collagen, which is Denisovan. And the jawbone looks. We think that Denisovans had very big teeth. We're not sure, right? And certainly this jawbone had very big teeth. It probably was Denisovan, but it's exciting and at the moment rather limited.
Okay, thank you very much.
Very good.